Hi, my name is Sid Gopalan. I'm in Todd Casto's lab in the Department of Biology. Today I'm going to be talking about how our lab studies the origins of evolutionary novelty and functional cell biology using single-cell sequencing methods. Our lab uses the snake venom system as a model to investigate a number of different questions related to how genes and the genome are regulated, how traits evolve, and how cells and tissues operate to produce extreme phenotypes. We were interested in understanding the cellular basis for the mass production of toxin proteins that normally takes place after a venom gland has been depleted, for example, after biting prey. Because this mass production may cause negative effects such as overloading the unfolded protein response and organelles in the cell that are tasked with properly folding and processing proteins, we wanted to understand how the snake venom system has evolved to potentially sidestep some of these constraints. In particular, we were interested in whether or not different toxins are produced in different parts of the gland, a term called heterogeneity, what causes this, and what regulatory networks are operating to allow these processes to occur without damage to the organ or the animal. To answer these questions, we decided to use single-cell sequencing methods. These are a family of technologies that allow for scientists to investigate elements of biology we'd normally need whole tissues to perform, such as measurements of gene expression and the accessibility of DNA. With single-cell sequencing, we can do the same things except on the scale of individual cells. These procedures provide benefits to us over traditional sequencing methods by massively increasing the effective sample size and sensitivity of our measurements. We first performed single nucleus gene expression measurement on a rattlesnake venom gland to begin answering some of our original questions. We first used our data to investigate the potential role of space limitation in generating heterogeneity, because genes need a lot of machinery to be made into protein that take up space. When genes are closely packed together, there may only be room for a few genes to operate at once. We found that for one small venom gene family in particular, the PLA2s, this seems to be a convincing argument. Separately, the activity of different gene regulatory programs can also drive cell differentiation and produce different gene expression profiles between cells. We first identified all gene regulatory networks whose activity differs between cells and quickly found that they were mostly factors involved in two cellular pathways, ERK and UPR, which are these conserved vertebrate cellular pathways broadly associated with cell growth, differentiation, and response to cellular stress. Investigating these two pathways more closely, we found that venom expression strongly correlates with the activity level of these two pathways in a coordinated manner, which tells us that their joint activity is the variable which activates certain genes at certain times. Part of the utility of single-cell sequencing is also the ability to combine data from different sources and technologies to improve on inferences. We took prior data on DNA accessibility and layered on top our single-cell gene expression data to make detailed maps of how every venom gene is regulated, starting with transcription factor binding. Our data also allowed us to identify the single most statistically important transcription factor for every venom gene, which is a question often asked by scientists for any gene, though often with lower power due to smaller sample sizes. Using a different technique, we combined gene expression data with DNA accessibility data from the same cell to identify transcription factors and where they bind the genome. This information also provided us much more power to infer high-quality regulatory interactions that help to partition toxin expression across the tissue. Bringing this all together, the evolution of new traits requires existing regulatory programs to accommodate the function of new genes. Our results suggest that existing pathways are particularly relevant raw material for this co-option to occur, which helps avoid constraints that would ultimately damage the cell. Single-cell sequencing is also valuable for testing the generality of these evolutionary strategies and holds exciting potential to broaden our understanding of the proximate and ultimate causes of cellular heterogeneity.